Yeah, so I'm going to present the work of many people who've worked on this in EBRAINS over the past few years. Yeah, so we heard this morning about data in EBRAINS, atlases, integrating data in atlases. And we heard a bit about one kind of model you can build with such data, the work presented by Victor Schasser and Ritter. I'm going to talk more generally about modeling and simulation in EBRAINS, in general doing computational neuroscience. So this slide shows a general modeling and simulation life cycle. So we have data from experiments, we have structural data, we have neuronal activity data. Using structural data, we can build models, run simulations, analyze the results, visualize the results, and then we can compare the results that we get from simulations to the results we get from experiments, physiological experiments in animals and in humans, and compare the model to the data and validate the model against the experimental data. So EBRAINS aims to provide tools for all of these parts on the right: model curation, simulation, analysis, visualization, and comparison and validation. But why? I mean, people have been doing this for many decades now. So why use EBRAINS for this? There are many challenges in computational neuroscience. Lack of compute power is increasingly a challenge. As we get big data, we can record from more and more neurons with neuropixel probes and so on, increasing the resolution of like 11, 12 Tesla MRI. So the amount of data coming in is larger, and so we need more compute resources to work with that data. Similarly, there's a move towards more sophisticated models, so addressing multi-scale simulation challenges where we want to integrate data potentially from the molecular level up to the whole brain level. And there again, we need more compute resources to simulate these more complex, more ambitious models. Another challenge that kind of follows on from that is that as our ambition grows, our software, the capabilities of our software grow, but concomitantly the software's complexity grows. And there's a general problem that these things become more and more like black boxes. We see this with machine learning, these very large artificial intelligence models. Understanding what's inside the black box becomes more and more difficult. A second problem is that a lot of these tools are developed by students, by postdocs, but in general, people who don't have a lot of experience in software engineering. They don't have formal training in software engineering. And so you can ask what the quality of these tools is, and the quality is quite variable across the field. A third challenge is the need for collaboration. Again, since we're now starting to take data from many different parts of the brain, we're discovering that we get a lot more from multidisciplinary approaches. And so we need to get people from different fields with different expertise. Even within a field, people are increasingly specialized. And so multidisciplinary can even be within a field these days. So we need tools which can help with this collaboration. And finally, a big challenge is reproducibility. Although it's a computer, so it's a computer program, in principle, it should run the same every single time you run it. However, these workflows get increasingly complex. They have an increasing number of dependencies. You need this library in this version, and this library in this version, otherwise it doesn't work. And over time, things just stop working. So some data analysis which you ran in the first year of your PhD, the odds are if you try to run it again in the third year of your PhD, it won't run because something's changed. You've upgraded your operating system. You've got a new version of some key library. So how can EBRAINS address these challenges? For compute power, EBRAINS offers the EBRAINS lab, which is a Jupyter lab hosted in HPC centers across Europe. There are federated high-performance computing resources made available from sites in Switzerland, Germany, France, Italy, and Spain through the Phoenix program. And there are these novel neuromorphic computing systems, which Catherine Emmons introduced you to this morning. On the topic of software complexity and software quality, well, one thing we can at least try to find software which is useful for us by going to the knowledge graph described in the previous session by Jan Bialy. EBRAINS is putting in place quite strict software quality standards. So this problem, a lot of software developed without much software engineering experience and by people who perhaps move on after a few years. So EBRAINS as a whole has this, or HPP as a whole has this software engineering experience, and this is being propagated out to the different labs developing this software. So the quality of EBRAINS software should be much higher than the average for the field of computational neuroscience. And one way we do that is by testing everything using continuous integration. So anytime there's a new version of the code, it's tested, check that all the old things still work, as well as the new features we've just added. For collaboration, we have many tools. I didn't mention here perhaps the EBRAINS community website, but also in particular the EBRAINS Collaboratory is designed as a collaborative workspace where people can come together to do real work on the same project, easily share data, easily share code in a controlled, private or public way. And for reproducibility, so EBRAINS is developing standardized workflows. We'll hear more about that in a parallel session later this afternoon. And EBRAINS is standardizing its releases of software. So all the software in EBRAINS doesn't only get tested on its own, but is now getting tested in connection with other software. So if you need 3 different pieces of software to do a certain workflow, we are testing that those 3 bits of software work together. And so every 6 months or so, there's a new version of the EBRAINS software library is going to come out. So you can be sure or much more confident that all the software in that version of the library will all work together. So I'm now going to go very fast through the different, oh, sorry, no, not at that stage yet. A bit more on the hardware before I get to the software. I mentioned the lab. This is who here in the audience knows what Jupyter notebooks are? Yeah, okay. So the lab is a hosted Jupyter lab. Essentially, it means that, so for anyone who doesn't know, Jupyter notebooks are a nice way to present, to run code together with explanations in a kind of logical order. It means you can develop and run programs in the cloud from your web browser. You can install any software locally on your own machine. And so we have these EBRAINS tools and software are installed in this lab environment. It runs on the HPC centers, and it gives users a free Jupyter notebook, which they can use for their research. The advantage of this compared to other solutions like Google Colabs, for example, or Binder, is that you have all the EBRAINS software ready tested, ready installed into the environment. So you can just get up and start working straight away. In terms of the HPC resources, so there are 5 European supercomputing centers are involved. They federated their services and making some of their resources available to EBRAINS. Almost all EBRAINS resources are now running on this so-called Phoenix research infrastructure. Because it's very easy to move data between the different sites and to run the same workflows on different sites in different countries. And then finally, we have these novel neuromorphic systems very much inspired. So rather than like traditional computers, these are very much inspired by the architecture of the brain itself. The brain scale system is based on mostly analog, mixed signal, but largely analog models of neurons and synapses. Then with digital connectivity, it runs very, very fast. This system is available in Heidelberg, but available across the 2 EBRAINS in the cloud effectively. Then the Spinnaker system in Manchester in the UK is still a digital numerical model, but very, very, the architecture is designed specifically for neural simulations, and so it can run much faster than the general purpose supercomputer. And you can access both of these through EBRAINS. Yeah, so moving on now to the different software tools available in EBRAINS. There's a large number, so I'm going to go very fast through a lot of slides. There's no expectation that you remember any of these, but you might spot a tool which you think would be valuable for your research. I will make the slides available at the end, online and on the website after the workshop, so you can come and you can, the tools which interest you, you can take the time to go through them in detail, and we'll do a very quick overview. There's just a question: how do you find the right tool? There are many different ways you can categorize tools in computational neuroscience. I'm going to structure it mainly by the workflow stage, so model building, then simulation, then data analysis, then visualization and model validation. But of course, you can also classify tools by what's their scope. Are we studying like molecular aspects of neuroscience, or single neurons, or networks, or the whole brain? The talk, by the way, does not include bodies. So the brain is in the body, which interacts with the environment, but that will be for the next talk on robotics. So I don't cover that here. And then there's also different levels of abstraction. So you can be studying single cells, but you can be studying them at the molecular level and studying how molecules move within cells. And so, and similarly with spiking neural networks, you can have simple point neuron models, or you can have very detailed, morphologically detailed neuron models in your networks. And so on each of these slides, there are these various icons which show you which of these different aspects that tool is addressing. So first of all, tools for building models. We have RDoc, which is for at the protein level. We have the molecular level tool set, again at the molecular level, and this, just to note, is a so this, the cogs on the top right hand side means that this is a programming tool. If there's a screen, it means it's a visualization. It's a web-based tool. We have CNRS ligands. GROL is a database of modulators of synaptic receptors. We have a subcellular web app which lets you create and build brain molecular networks. A tool for extracting features from recordings, like the height of the spike, the width of the action potential, and so on. A tool for building Hodgkin-Huxley-based neural models in your web browser. A suite of tools of libraries which let you build in a principled workflow, biophysically detailed, biologically constrained neural models, and compare them and validate them against experimental data. A tool called Pine, which is from my lab, which lets you build simulator-independent spectrum neural networks. They run on different simulators with the same code. There's a hands-on session on Pine later on this afternoon. You have NestML, which lets you build ion channel models, or different spiking neural models, or different synaptic plasticity models, and then use them in the Nest simulator, and in future also in the Spinnaker system. You have Snuda, which is a tool for building networks of biophysically detailed neurons, in which the connectivity is based on the neuron morphologies, so detecting where dendrites touch, for example, tells you where the synapses can exist. The brain scaffold builder is a somewhat similar tool, but also lets you combine both detailed, morphologically detailed neurons and point neurons. The virtual brain we've heard about already, so I won't go into that in any more detail. A suite of Jupyter notebooks for single cell modeling workflows, and that's for building models. For running simulations, we have many tools. At the nanoscale level, molecular dynamics simulations. This is an example where this work was used as part of the COVID-19 response to investigate the impact of COVID-19 on the nervous system. Diffusion of proteins, a web-based tool for running simulations of single cells in the web browser in real time and seeing the results directly on your screen. a more complex tool where you can run experiments, virtual experiments with small circuits, in this case of the hippocampus of the rat. You can choose where you put your virtual electrodes, where you record, and parameterize the structure of your recording. And then a much larger scale tool for a full scale model of the entire rat hippocampus CA1 region, again, running in your web browser, but in reality on these EBRAINS resources behind. You have the NEST simulator, which was mentioned already this morning. Very scalable, runs on HPC machines up to very large sizes of networks. Arbor, which is a tool going in the same direction, but with for detailed, morphologically and biophysically detailed models of neurons rather than point neurons. You have the classical neuron simulator, which has been enhanced within HPP and can be used for both these types of networks, so both point neurons and morphologically detailed, biophysically detailed models. And the nice thing about having these different tools is you can cross-check. You can check that you get the same results with neuron and with Arbor, for example. If you want to use the neuromorphic systems, Brainscale and Spinnaker, we have an interface for that. So you can write your network description using Pine, which I mentioned before, and then send it off to Manchester or Heidelberg to run the simulation and get it back on your local machine. In development, a multi-scale co-simulation framework to let you connect these different tools, not just in sequence, but actually at the same time. So for example, you could have part of your network running with simplified point neuron models in NEST, and another part running in with more detailed neural models running in Arbor, and communicating in real time, sending action potentials backwards and forth between the two simulators. Again, more simulation tools for molecular dynamics, and so those are the tools for simulation. Moving on to data analysis, we have a whole suite of tools for data analysis. I'll go through, I think I'm running out of time, so I'll go through rather quickly. LFPy is a tool for calculating local field potentials and potentially EEG from your simulation results. We have tools for visualization at different levels. Again, as I say, don't worry, you can't keep up with this. The slides will be available afterwards. And then finally, we have tools for validating models against experimental data, a suite of libraries which help you structure this, this comparison, formalize this comparison, and then a catalog of models and tests which have the results of comparing these models to these experimental data sets. And then putting all these tools together, EBRAINS is developing systems for computational workflows based on CWL, the common workflow language, and linking back to the knowledge graph, which we heard about earlier. All the results of these computational workflows can be captured and stored in the knowledge graph. And so when you come to publish the results of your analyses or your simulations, that information is already there in the knowledge graph in the private space, ready to be made public, to be curated and made public. And there's a hands-on session on that also later on this afternoon. So in summary, EBRAINS offers many hardware and software resources for modeling and simulation, from molecule up to the whole brain. EBRAINS focuses on software quality, on reproducibility of simulation results. All of these tools are open source. So if you start working with a tool and you find a bug, or you have an idea for a new feature you'd like to add, you can go to GitHub, you can get involved in developing these tools yourself. To really push reproducibility, every 6 months there will be a new release of the new versions of EBRAINS tools, all tested so that they actually work together, interoperable. And EBRAINS is providing automated workflows to let you combine all of these tools together. So thank you all. This work was performed by many, many hundreds of people, certainly over 100, way over 200 people in the Human Brain Project across many institutions. And I haven't got space to thank them all here, but you can see the individual contributions if you follow the links on the slides. If you have any questions, my email is at the bottom of this slide. Thank you very much.